Hi, good morning. Here for my place is already afternoon. My name is Lihua Fan. I'm working at Kentville Research and Development Center. First of all, thank you for the organizers to organize this research day, and also thank you for these opportunities. And I would like to present the highlight of AAFC CAP research result of activity 13. This research is funded by the Canadian Agriculture Partnership AgriScience Cluster Program. The overall goals is forcing sustainable growth of Canadian grape and wine. Our research theme is Activity 13, that is a native yeast and wine quality. The participants include AAFC researchers from Kentville Research and Development Center and the university researchers from Mount St. Vincent University, San FX University, and Acadia University. Due to the limited information regarding the yeast communities in Nova Scotia vineyard and the fermentation properties of native cool climate yeast, the objective of our research was to identify and characterize native yeast strains, conduct sequential and mixed fermentation using selected yeast strains together with traditional Saccharomyces cerevisiae, determine yeast quality parameters, and conduct sensory evaluations of the resulting wine. By far, more than 300 yeast strains were isolated, identified, and stored in our lab for our long-term research use. Yeasts were characterized for their fermentative properties, including alcohol and sulfur dioxide tolerance test, hydrogen sulfide production, different enzymes activities. The database was created for our selected yeasts. This slide shows that yeast tolerance tests were determined using a bioscreen C instrument and microplates with 200 wells. That will generate numerous growth curve of the yeast. So based on that result, we will conduct a statistical analysis based on log, lag stage, slope of the curve, and highest OD values. So a case study on yeast communities before and after spontaneous fermentation was conducted. This research has been published in the Canadian Journal of Microbiology. In this study, the yeast communities of Price Lacady Blanc grapes were characterized by PEG biosequencing. In this graph, you can see that light blue colors part here that represent the Saccharomyces yeast. We found that species of Saccharomyces yeast only made up 0.04% pre fermentation. That is pre fermentation. And then that will reach to 85% to 100% after fermentation. That is the part. Okay, so a better understanding of yeast communities allow us to make better use of non-traditional yeast and spontaneous fermentation to produce wines unique to the regions. During 2018 to 2020, Lacady Blanc grapes were harvested from 3 organic vineyards. That is the first one, second one, third. And then we have harvested 5 conventional vineyards. That is represented here, that blank box. So we studied these yeast communities. Our results showed that there were marked differences in the yeast species, mainly related to the non-fermentative yeast between organic and conventional vineyard. This work helps us to interpret the complexity of the vineyard ecosystems, suggesting that cultivation practices play an important role in the microbial terroir of hybrid grapes grown in the cool climate region. This research also published in the Journal of Applied Microbiology and listed as an editor choice. For our fermentation trials, the yeast strains were selected including Saccharomyces yeast, for example, Saccharomyces yeast euverum, and non-Saccharomyces yeast. Mixed and sequential fermentation trials was conducted. The fermented wine sample were collected for quality analysis, such as sugars, organic acid, phenolic compounds, and volatile compounds. Sensory quality of fermented wine products were also evaluated by our trained panelists. Our fermentation research has been published in the Plants, People, Planet. In the following slides, I will just give some examples about our quality evaluation and sensory evaluations. This table is an example of quality parameters of wine fermented with our selected non-traditional yeast, inoculated on day 1, and then followed by inoculate or commercial yeast of Saccharomyces cerevisiae that inoculated on day 7 or day 14. We found that the total phenolic compounds of the wine were not significantly different among all samples. However, alcohol levels were lower, ranging from 8.9 to 10.2 compared with the control of 11.3%. Glycerol level did not differ, but fructose levels were higher and the total acidities levels were lower compared with the control. This slide shows the correspondence analysis of sensory evaluation data from the sequential fermentation, suggesting that sequential fermentation using our selected Saccharomyces euverum and non-Saccharomyces yeast were associated with a fruit attribute. As you can see the different, this part is the control, and this part we can represent some fruit attributes. So this with more for our fermented samples with more appealing characteristics of citrus, apple, sweet, pear, floral, or tropical fruit. The timing of the sequential fermentation with Saccharomyces yeast control, for example, day 4, day 7, or day 14, did not appear to influence the flavor profile of the resulting wines. Our results demonstrated that native yeast selected from cool climate wine regions can be used in concert with traditional wine yeast to produce novel, lower alcohol levels wine with a low risk of fermentation failure. So based on our research on yeast communities, based on the spontaneous fermentation or after fermentations data, as well as literature research, we have just recently completed a review paper regarding non-fermentative yeast of wine grapes and their potential application to winemaking. So this paper is under review by the Journal of Comprehensive Reviews in Food Science and Food Safety. This slide shows our technology transfer effort. Over the past few years, we have given numerous presentations at scientific conferences and industry meetings, including Canadian Institute of Food Science and Technology, the Grape Grower Association of Nova Scotia, the Canadian Botanical Association meeting, Canadian Fungal Research Network meeting, the International Cool Climate Wine Symposium, International Association for Food Protection. And last year, we gave the presentation at the Federal Food Safety and Nutrition Forum. We also trained one PhD student. She got her PhD title last year, and also 3 co-op students from Acadia University. So I would like to use today's opportunity to acknowledge Canadian Agriculture Partnership AgriScience Program, Canadian Grape 1 Certification Network, GGAN, Nova Scotia Grape Growers, our researchers at Kentville Research Development Center, and our university collaborators. Thank you very much. Thank you for your attention.